Good morning, good afternoon, and good evening, everyone. Welcome to today's webinar co-hosted by the Boston chapter of the American Statistical Association and Cytel, featuring Dr. Frank Bretz from Novartis. I am Gauthier Po, Biostatistics Team Leader at Sanofi and the Program Chair of the Boston chapter of the ASA, and I'm absolutely delighted to welcome you all today for our first webinar of the year. So before we begin, I would like to take a minute to introduce the Boston chapter of the ASA. We are actually one of the largest and most active ASA chapters covering Maine, Massachusetts, New Hampshire, Rhode Island, and Vermont. Our community brings together more than 850 statisticians and data scientists from academia, industry, and beyond, fostering collaboration and knowledge sharing. Our mission is to advance statistics and its application by providing educational and networking opportunities. To achieve this, we host a variety of events throughout the year, including webinars, lectures, short courses, award dinners, and social gathering. Our main event is the Boston Pharmaceutical Statistics Symposium, usually held in the fall, which gathers more than 200 statisticians from Boston area and beyond. So here is the current list of our planned 2025 events, with a busy first half of the year with webinars, conferences, and an award reception. After a summer break, we will be back with more events, including one ASA traveling course and our flagship event, the Boston Pharmaceutical Statistics Symposium. We add new events during the year, so please make sure to check our website for the most up-to-date information. Our next webinar is on May 6, featuring Dr. Wei Wei from Yale. Registration information will be shared later. So we welcome anyone who wants to join our chapter, regardless of your location. So you can join either through your ASA membership or directly to the Boston chapter. Feel free to reach out to me for more information about the chapter, or if you're interested in taking an active role in one of the events, or if you have new ideas to bring to the chapter. So before we get started, I just want to share a few logistical details. Please note that today's session is being recorded, and the recording and slides will be shared with all registered participants after the event. And we encourage you to ask questions, so please submit your question using the Q&A Zoom feature at any time during the session. We will monitor the questions during the webinar and address them during the dedicated Q&A at the end of the presentation. So without further ado, I will hand it over to Yanis Jamier from Cytel, who will introduce our speaker today.
Thank you, Gauthier. And good morning, good afternoon, everyone. Thank you for joining us today. My name is Yanis Jamier. I'm the Chief Scientific Officer at Cytel. And we're thrilled to be co-hosting this webinar with the BCASA. And it is absolutely my great pleasure to introduce our speaker, Dr. Frank Bretz. So Dr. Bretz, if he should be well known to many of you, is a distinguished quantitative research scientist at Novartis and a leading voice in pharmaceutical statistics. His extensive contributions span adaptive designs, dose finding, estimands, and multiple testing. He also holds adjunct professorial appointments in both Germany and Austria, and has served on international regulatory working groups, including ICH E9R1 and E20. Today, he'll be presenting some joint work with other colleagues that I'm sure he'll mention, generating the right evidence at the right time, where he'll introduce FACTIVE, a flexible clinical trial framework designed to generate high-quality evidence that meets the needs of diverse stakeholders across the healthcare ecosystem. With that, please join me in welcoming Dr. Frank Bretz. Over to you, Frank.
Okay, so thank you so much for the very kind introduction and invitation to give this presentation. Just a quick check that you're seeing the slides.
Yes, we do.
Thank you. Okay, so it is really my great pleasure to give this talk today. But before anything else, I must acknowledge that I have heavily borrowed material and slides from Cornelia Dunga-Baldauf, Anja Schiel, and Nigel Yateman. So many thanks to them for offering their slides, but of course the views are entirely my own. So this talk is based on a paper that appeared about 1.5 years ago, and the work was led by Cornelia. She actually might have joined the seminar today, and if so, she will hopefully chime in if I miss anything. So this collaboration is a joint work with several colleagues, among others, Rob Hemmings, formerly one of the lead regulators in Europe, Anja Schiel from the Norwegian Regulatory and Reimbursement Agency and one of the leading voices on the health technology assessment side here in Europe, and Chris Holmes, professor for statistics at the University of Oxford. Now, in part, this paper and therefore the presentation I'm going to give was triggered by a new regulation on health technology assessment that came to effect literally just a couple of months ago, beginning of this year. And I will talk about this new regulation in a minute and how it triggered the type of work that we are presenting. Now, the methodology we are proposing, that is this framework that we call FACTIVE, and the underlying principles hold much more generally. So the way I'm going to motivate the methodology takes the European jurisdiction as an example, but all the methodology, the framework would hold much more generally, and I will make specific connections to the US system as I walk through the presentation. So this is therefore the outline. I will first give a somewhat longer introduction from approval to access, because there is this new regulation in place here in Europe. It would be good to understand this. Coupled with that, I have a motivating example, which I assume will also reflect some of your experiences that we have made. And these two sources, so to speak, they triggered this FACTIVE methodology. And that's our proposal, so to speak, to address those changes and the issues that we have seen. And then I will conclude with a short discussion. So how do we get from approval to access? So this slide would reflect the three different stages: approval, assessment, and appraisal. So approval is the usual setting of clinical drug development, the clinical trials that we are running, designing, and analyzing in order to collect the clinical evidence with best or at least high internal validity in order to have good understanding of the benefit risk of the treatments that we are developing. Then at the assessment stage, we'll have a quantitative summary, so to speak, and a review of the quantitative information and evidence synthesis step in order to understand the value of these treatments. Now, this is not a qualitative assessment. And for example, at this stage, there's not even said whether A is better than B. But this really then comes in at the final stage around appraisal. This is where the information, all the evidence has been gathered, will go into the health economic decision whether to pay or not to pay a specific drug or treatment. And here's where external validity plays a very important role. So these three stages, of course, they come with different mandates. So on the left-hand side, you see the approval, which of course involves interactions with regulatory agencies such as the FDA and EMA. Here is where we are generating the evidence using clinical trial frameworks under GCP, under these GCP connections, umbrellas. That's where the ICH guidelines kick in. So it's all this where we generate information in order to characterize the best, the benefit risk of our therapies. Now, then this evidence being generated, that will then be helpful for the evidence review and synthesis at the assessment stage in the middle. And this is where we have the interaction with health technology assessment bodies. Here in Europe, assessment is typically done on a national level. However, as of this year, the Joint Clinical Assessment, for short JCA, has been put into place. And this JCA takes place exactly at the transition from approval to assessment, as I will explain on the next slides. Now, once the assessment is completed, then the next stage is the appraisal, which involves reimbursement decisions. And to be honest, that's a world on its own where I have personally really no experience at all. But It is important to understand that from left to right, the information that is, so to speak, the information flow, but also we have at the very right is we have the key decisions when it comes to reimbursement decisions. And you may also notice here on the slide is that the error of framing key questions does not go all the way from right to left, meaning that the evidence generation is mostly dominated by regulatory agencies. So this will lead to the different sets of questions and requirements that these different stakeholders will have, as I'm going to show in a minute. But let me come back a little bit to this new regulation on health technology assessment here in Europe that I had briefly mentioned. So as of January 12th this year, you can see that we have started a transition period of 5 years into this new model under the Joint Clinical Assessment, JCA. With that, we have a new set of rules that are in place that will apply to companies seeking marketing authorization for their products by introducing a new and permanent European framework for health technology assessment, introducing a single European level submission file for joint clinical assessments. And it is good to understand that this market authorization, or in other words, the submission, and the JCA, this joint clinical assessment, these are two processes that go hand in hand and literally tied to each other. So what you see on this slide, in the upper panel, you see the timeline for a submission process in Europe. And in the bottom half, you see the JCA timelines, and they are intertwined. So a submission to the European Medicines Agency, the European Health Authority body, that would trigger the joint clinical assessment. So after CHMP adopts its questions at day 120, JCA will finalize its assessment scope via the PICO framework. And there's an expectation that 100 days later, sponsors will submit their dossiers. So I use the word PICO that, and you see that on the slide. So PICO is abbreviation for population intervention, control, and outcome. So, and this is a framework that has been used by the HTA bodies since more than 10 years, maybe 15 to 20 years. And it's conceptually very similar to the estimate framework in the senses that allows a precisely crafting the questions, formulating the questions that are of interest. So the JCA will start finalizing its assessment scope via such a PICO framework, pass on their questions in using the PICO framework to the sponsors, and then sponsors have about 100 days to come up with their supposed answers via their dossier. Now, 100 days is really a very short time, and I think there's a very strong expectations that sponsors will be well prepared and not just start thinking about the questions once they receive those the PICO questions. With that said, the PICO will not fall out of the blue, and sponsors being well prepared will have to think about what could be potential questions that such an HTA body might ask, specifically the JCA. And I mention all this because this is what has been introduced in over the last few years, and that's which triggered the work on FACTIVE, as it would facilitate the thinking process early on so that sponsors have thought about the questions that they may receive and have generated the necessary evidence. And keep in mind that this JCA operates in close connection, at least time-wise, with the submission here in Europe with the regulatory agencies like EMA. So on this slide, you see on the left-hand side, again, these timelines side by side. On the left-hand side, vertically ordered. And on the right-hand side, you see the formal process for this JCA. So it starts with the JCA process all the way to the finalization of the JCA report. I'm not going to go into any details, but just to say that this is indeed a formal process, and it has a legal framework, a binding legal framework as basis. And that is the screenshot on the top right. So this would be a legal framework like the 21st Century Act in the US or a pharma legislation in Europe. And is accompanied at the bottom with an implementation regulation, which is there to understand, which provides explanation how the legal framework should be implemented. And this implementation of regulation then comes with a series of guidelines specifically for this JCA process, such as the procedural guidance, the appointment of assessors, guidance on scoping process, and, for example, guidance on JCA dossier. And there are other guidelines. So you see we have now this formal process in Europe, which puts the market authorization / submission side by side with the joint clinical assessment. And not surprisingly, the questions that you might get from different stakeholders could be very different. So on the left-hand side, you see, of course, our common domain in the world of clinical trials that are being optimized to minimize heterogeneity and maximize treatment effects to ensure causal conclusions. Causes in the sense that a clinical trial is designed so that there are very few alternative explanations left in order to explain a beneficial effect due to treatment. And in particular, we would use placebo or perhaps one active control. And all this leads to the usual estimate concept that you all know very well, what the clinical trial is intended to estimate. On the right-hand side, here is where you see that how the population is put into a quote-unquote real-world context, leading to the PICO concept that is very familiar within the HTA community. And it is important also to understand that clinical practice is very different in different regions and countries. So as a sponsor, however, we need to provide the evidence for both sides, offering a path from the left to the right, as opposed to the current split, at least at my company, of having clinical trials on the left, followed somewhat independent by the real-world studies on the right. So what I mean by this is that we need to support informed decision-making to provide clear descriptions of the beneficial and harmful effects of a treatment to various stakeholders. But different stakeholders, such as regulatory and reimbursement agencies, may have different requirements. Now, the current paradigm is that we would generate this evidence sequentially through market approval all the way to access. So sequential means that we have a sequential series of studies. And however, data that's being generated later in this sequence, perhaps through observational studies, can be difficult to compare with earlier randomized data. And I have such an example in a few slides where we show the difficulties in interpreting the results. So this leads to all sorts of difficulties in understanding and interpreting the treatment effects. Moreover, the scientific questions these later experiments can serve to answer often remain vague and may not address stakeholders' needs. Sorry. So what you see on this slide is now a visualization of this sequential approach that that's what we have done in the past most of the time, that we have our conventional phase 3 program displayed here in green. Then we have this white space where we certainly talk to regulators. We might have some conversations with payers, but often we have a series of other post-marketing studies afterwards, and that's a mixed bag as indicated by the different colors, green and yellow and blue. Now, what you also see is on this timeline depicted below that at some point we get the approval, and somewhat later we have the first availability of the new treatment to patients, and that's indicated by this little dot 1. And at that time, this is where we have the initial uptake of our new treatment use. And of course, as in these treatments becoming, getting more shares in the market, then there will be a maximum uptake of the new treatment use at some point. Now, what is understood that this white space has been relative constant for almost, for more than 10 years now. And what you see here is a number quoted by FPIR, which is the European analog to the pharma. So it's the European Trade Association of Pharmaceutical Industries. So, and according to FPIR, you see it's this white space may easily take like 1.5 years or more. And it has stayed roughly the same over the last 10 plus years. And this is also because at the time of regulatory approval, key information needed to support payer decision is often not available. That would include data on resource utilization, patient quality of life, long-term safety, and effectiveness data, and so on. And here's an example to illustrate this very last point, that often we may have conflicting data or difficult to interpret data, and we don't have the right data for payers' decision. And this is an example from internal. So it's Lucentis, one of our compounds, and I'm using here an example for a certain eye disease, namely neovascular age-related macular degeneration. And in this particular setup, we actually do have 2 double-blind randomized controlled phase 3 trials, Marina and Anchor, which investigated the efficacy and safety of Lucentis after 12 months of treatment. And this 12 months of treatment would consist of monthly injections of the compound into the eye. So it's a very inconvenient procedure, but that's essentially what that treatment, the series of interventions is, monthly interjections. In addition, we have a post-marketing real-world evidence study, Luminos, which investigated the effectiveness and safety of Lucentis at month 12 in form of a single-arm trial in treatment-naive patients. And importantly, the treatment patterns would be determined under real-world conditions. And what I mean by this is that we would not have necessarily monthly injections, but the injection frequency would be per discretion of the treatment physician. So the treating physician could decide on, for example, that injections are only needed, would only be given on an as-needed basis, and therefore lead to many less injections possibly. So here you see the efficacy results or effectiveness results of Lucentis. So what's being measured is the visual acuity in terms of increase in the number of letters based on the standardized charts that patients are going to read from after receiving the treatment. And in the 2 RCTs, we saw an average improvement of 7 and 11 letters, whereas in the real-world study, mirroring clinical practice, we only saw 3 letters of improvement. And of course, this led to a number of questions. Why we have these discrepancies and what is happening in the clinical practice, and foremost, how we can interpret the results of these 3 studies. We certainly have a different patient population, although and we certainly have different treatment conditions in clinical practice. I mean, there's, of course, in the clinical practice study in Luminos, we have no blinding. We do not even have randomization because it was a single-arm trial. In addition, we had partly different assessments in the clinical practice because different charts were used for letter reading and to assess improvements. And of course, we had different treating schemes. We had, on average, much fewer injections, not 12 per year, but many less, depending on the different treatment schemes deployed by the treating physicians. Even more so, complicating the situation is depending on the reimbursement schemes or other administrative conditions, there might have been a delayed start of treatments for individual patients, depending on in which regions they would be treated and the treating physician would be in. So we have to be aware, of course, that the comparison across these studies that we have is not a randomized one, and there are several years apart between the 2 RCTs and the Luminos study. Now, we could have done some population matching, and in fact, the inclusion-exclusion criteria were rather similar in all these studies, but inclusion-exclusion criteria is not everything. And we could easily show that the population was still somewhat different. And then, of course, new treatments for the disease had also emerged in the meantime. So even if we had matched the populations, we would not have to, we still would have to make important assumptions and unverifiable assumptions, such as no unobserved confounder, in order to support any causal statements. Now, the end of this particular discussion is that we had difficulties in talking to our HTA bodies, and the main interpretation conclusion of these results was rather qualitatively rather than quantitatively, is that the one conclusion drawn is, and that's probably a very plausible one, was not to delay the start of treatment after a diagnosis and treat sufficiently often. But still, this exercise then also made us wonder whether we could have taken a different approach. So we wondered whether we could have collected the data in clinical practice much earlier on in order to answer the HTA requirements and questions. And then if you want to compare clinical practice with RCT conditions, how we could possibly draw reliable conclusions and how we can therefore collect the data in a structured way. And what can we do before registration or approval, as the drug will not be yet on the market. So these were, so to speak, the questions that we had in mind, the guiding questions, which led us to come up with a proposal that to move away a little bit what we did in the past, this purely sequential approach, to what we might want to do in future. And what you see here is that we suggest augmenting phase 3 trials, the development program in phase 3, augmenting it by including certain close to real world elements that would be important to collect that information to collect early on. And so this is depicted by the shift of the information that's depicted in blue, that otherwise we would have collected in post-marketing studies, but now we are suggesting to collect it as part of an augmented phase 3 program. And the hope would be, as you can see in the purple shape, that by collecting this information early on, that would convince the payers to make quicker and better reimbursement decisions. Quicker in the sense that the time to the first availability, so the number one dot, would occur quicker, faster, sooner after the approval, and the initial uptake of the new treatment use, so this is the number 2 dot, that this is a somewhat larger and overall leading to a larger maximum uptake of the new treatment use. So that's what we are hoping for. Now, the question is, of course, then how we would augment such phase 3 trials in a reliable way. And this is, so to speak, the rest of my presentation. So this is the proposal for flexibly augmenting our clinical trial designs for improved evidence generation. The idea is that we would augment our current phase 3 trials under one single protocol and, very important, without touching the core RCTs. And what we mean by all this is as follows. So we would first understand that there is a broader population that we might be interested in. Broad in the sense it goes beyond the population that would satisfy the regular RCT entry criteria. So we would start screening patients which we expect will be treated in clinical practice. This patient population is comprised of the patients in green who fulfill the usual RCT criteria. That is a narrower population where we want to demonstrate efficacy with the advantages and benefits of our usual RCTs for registration. And the patients in blue who do not fulfill all these criteria. Now, in doing so, of course, we would exclude any patients, for example, where the product could be a safety risk and so on. So, of course, we would not include these patients. But we have this concept of the broader population. Now we can start designing our core RCT. This is the regular RCT that's so that patients in green, the RCT eligible patients, they would be assigned to this core RCT and randomized to either treatment and control as usual. So this is the regular phase 3 trial, and we would not touch this part. It's the same population, the same treatments being compared, the same endpoints, and the same sample size. But then we would augment this core RCT. And one way of doing this is to, for example, wait for an interim analysis. And if that one has promising results, we would then start another part of this trial which has close to real-world elements. Okay, this is what we call CRW, and we want to kind of illustrate with this term close to real world that this is not the true real world, in the sense is it's still part of a clinical trial after all, right? So it's not real world. But from now on, we would randomize the green patients, the RCT eligible patients, to either the core RCT or the CRW part. And within the CRW part, we would then randomize the patients to either treatment or control, but under close to real-world conditions, such as more flexible treatment schemes, more flexible concomitant medications, less regular visits, and perhaps even different endpoints that we would otherwise not cover in the RCT. And finally, to complete this picture, we would also consider randomizing the blue patients to the CRW part, those that are not eligible for the core RCT, but we can still capture during the screening process, and we would then use these patients and randomize them to treatment or control under either RW or RCT conditions. Now, so this is the complete scheme. And of course, this complete scheme is fairly complex. And in practice, I would not see it being implemented with all the details that I have shown here. But the whole purpose of this scheme is an invitation to think about generating the right evidence at the right time for the different stakeholders to come, be it reimbursement agencies, be it regulatory agencies, or maybe other important stakeholders. So in practice, I would think that we probably will only use specific aspects of this overall design, depending on the specific context. And on the following slides, I will show a few applications. But before moving on, I just want to emphasize one more point that we have made in the supplementary material of the paper, which is goes a bit more into the actual statistics. Because by the virtue of this design and the way how we set up the randomization and all this, we actually are able to unbiasedly estimate a number of different estimates. So we show there a list in the supplementary list material. We actually show a list of 8 very different estimates. So this design not only helps to understand the collecting the right information, but it also helps in facilitating the discussion about what are the treatment effects we are truly interested in for different stakeholders. So for example, are we interested in a treatment effect under RCT treatment conditions for RCT eligible patients and the target population? Or are we interested in the effect of treatment conditions within the treated? Or are we interested in the difference of treatment effects between treatment conditions? Are we interested in the heterogeneity of treatment effects in the treatment under RCT treatment conditions? So there are many very different treatment effects we could be interested. And by virtue of this randomization and so on, you can easily imagine that you have a much better way of getting causal conclusions and answering those questions than if you have studies that are several years apart and which are very difficult to compare. So a few applications and some experiences that we have made. The first one actually goes back to the diversity-related documents that FDA started to release since the early 2020s. And the first one that got our attention was the one on enhancing the diversity in clinical trial populations. And in that guideline, FDA mentions the importance of broadening the eligibility criteria so that the study population better reflects the patient population likely to be used in clinical practice. And this, of course, is exactly what we have here demonstrated, like in the, what is here, you see that the little fence and the box, the red box around it. So this is exactly the broader patient population that we had in mind when putting the FACTIVE methodology together. And in fact, having this methodology in place, this framework in place, helped us a lot in navigating through the various FDA guidelines and understanding how we could possibly design our trials in view of their requirements using this scheme that you have here, that you see here on the slide. So, for example, the same FDA guideline continues writing that a broader participant group could be part of a secondary analysis. So that we interpret to be analysis of the part that I have now ring-fenced in red. And so this would be a sub-part of the overall scheme, and you would focus on this part of the CRW of your clinical trial, and that would be part of the secondary analysis and could help in addressing FDA's requests. Another application that we sometimes received is to perform just the part now in red, which represents a randomized pragmatic trial. So in other words, a randomized pragmatic trial falls out as a special case of this FACTIVE framework. And when I say randomized pragmatic trial, that what I mean is, for example, the EMA scientific guidance on post-authorization efficacy studies describe the objectives of a pragmatic trials as examining interventions under circumstances that approach real-world practice. And note that they use the term approach real-world practice, and we use the word close to real-world. And moreover, in that guideline, EMA indicates the need for assessing heterogeneous patient populations, possibly less standardized treatment protocols, and delivery in routine clinical settings, as opposed to the regular research environment that clinical trials take place. So, despite the existence of pragmatic trials for several decades, only a few of these studies, of course, have contributed evidence for regulatory decisions, but they can play a much more important role when it comes to other decisions for other stakeholders, such as for HTA bodies. And so this was also taken up by FBIR, and they had a position paper on randomized pragmatic trials to generate high quality real world evidence for regulatory and also reimbursement decisions. And they also refer to this FACTIVE design. And so you might be interested in following that link. So another question that we received was whether a pragmatic component can simply be run alongside the core RCT. And the answer to this is yes, you can do that. And you see that ring fenced now in red. And what the advantage is that you really have this concurrency in time. So if you run the trial as shown in red, that would allow us to compare the RCT treatment conditions versus clinical practice conditions for RCT eligible patients. Without this, we will typically have this time gap of several years between the RCT and the clinical practice studies, which would be designed and implemented and conducted completely like in sequence with years apart. And this would lead to all the difficulties in interpretation, as I have illustrated in the Lucentis study. And one concrete application of this particular suggestion happened rather recently, a couple of years ago, in the development of a new CAR-T. So the setting for this particular application was that we were ready to start a trial, a two-armed randomized trial, the regular RCT for a new CAR-T compared to a non-CAR-T control. However, it's just literally a few weeks or months before we could start with first patient, first visit. A competitor had received approval for their CAR-T exactly in this specific indication. So now this would not be possible anymore to randomize to a non-CAR-T, because now the standard of care would have became the approved CAR-T, or would soon become the approved CAR-T. And FDA pointed it out that we would not be allowed to run this study anymore as originally planned and also agreed. So that now, of course, asks the question, what can you do? The team did not want to run a head-to-head comparison versus the new CAR-T for a couple of reasons. And one important reason was also the availability of the new CAR-T. So our CAR-Ts, the manufacturing of CAR-Ts are very advanced. It's not so easy to manufacture them. So even from a logistical point of view, would have been almost impossible to run an RCT this way. Plus that in general, they didn't want to have a head-to-head comparison. So the next reaction of the team was, okay, let's propose a single-arm trial to FDA, and the FDA immediately rejected that idea, probably for good reasons. So, and then we were almost completing and shutting down the development for this particular indication for our new CAR-T until we had some further discussions, and FACTIVE actually, so our framework helped facilitating on these discussions. Well, can we now not think about using the close to real world element, more specifically the new CAR-T, so the approved CAR-T, that will become available under real world conditions. So can we now, of course, our new CAR-T is still not available under real world conditions, but can we now consider randomizing patients under the approved CAR-T schemes under real world conditions, but we would only include patients that are otherwise RCT eligible. So in short, we therefore designed a study where we would have a single arm of our new CAR-T for RCT eligible patients under RCT conditions, and we would randomize the same RCT, or from the same set or pool of RCT eligible patients, some of them to the pragmatic single arm, generating the real world data on the approved CAR-T. So this was in a nutshell the randomized two-arm study that we were considering. So you see that here again on the top left. And in terms of analyzing the data, we would primarily focus on our single arm for our new CAR-T hypothesis and use our regular test procedures for that. And then we would use the single arm real world data on the approved CAR-T to contextualize the results of our single arm new CAR-T. That would help us to estimate the treatment effect, but also to contextualize the choice of the efficacy threshold used to test for the single arm new CAR-T. Of course, we could also have considered going beyond contextualization and consider testing formally between these two arms if that was of interest. So all this is to say that this design, so having the FACTIVE framework in place, helped us to think about a very different design that the team originally did not think about. And it would also have, we would, in a very natural way, tick many boxes that would be associated with guiding principles for collecting high quality real world data, as you see it on the right hand side. So that would come more very naturally because we would have prospectively planned about such a randomized trial, two-armed randomized trial. Now this design, just a quick note, is of course more adapted to regulators than payers. Because the real world patients would still come from the RCT entry criteria, but we would still could also have considered a broadening population to include also subjects, from the broader patient population, to accumulate further data for reimbursement discussions and stakeholder discussions and decisions. So that all brings me coming to the conclusion. Please bear with me because I'm based in Switzerland, that I use an analogy of the Swiss Army knife, or a versatile tool. So a Swiss Army knife, you can use it for different functions and for different needs that you have in different situations. So it's a very versatile tool. And of course, it's a very popular knife here in Switzerland. Yeah, the analogy is that using this FACTIVE methodology, it helps you to actually think through various, the design and the questions and the data that you need in very different settings. So the last example that I've just shown was about enhancing our single-arm trial in contextualizing the information in a randomized setting. Then I mentioned previously the pragmatic trials, something that that's hopefully will play a more important role in the future in our regulation and importantly in our reimbursement discussions. Then I mentioned the connection to patient diversity and FDA guidelines in particular. Then, of course, all this we have developed in order to fulfill better the HTA requirements. If you think back about my introduction of the new regulation that is being now established in Europe. And there might be other applications of this methodology and many different settings where we could use elements of this overall framework. So we have introduced a new class of study designs that enable a flexible augmentation of confirmatory RCTs with concurrent and close to real-world elements to facilitate the estimation of treatment effects in the confirmatory part and other complementary treatment effects in the close to real-world part. And also to facilitate stakeholder use of the evidence that's relevant within their own decision-making framework. The benefits, of course, is that we are generating the data concurrently under one single protocol. We, of course, would heavily rely on randomization to ensure rigorous statistical inference. I had briefly mentioned the possibility of estimating unbiasedly very different estimates, and therefore greatly facilitating the interpretation of treatment effects as opposed to having run these studies in sequence with different years apart. And overall, that also, we believe, leads to an earlier evidence generation for health technology assessment, and hopefully that the product actually gets quicker to the patients in need. So the proposed augmented design therefore offers various opportunities to accelerate not the development, but the patient access of a beneficial drug. It is primarily an invitation to think upfront about the questions that need to be answered for different stakeholders and the data that we need to collect to answer them. And flexibility is key so that we can tailor the design to the project needs. And you have seen through these 3 or 4 applications that I have that I've shown that we can use very different parts of the overall scheme and tailor it to the needs for our individual projects. And lastly, just to close the loop to the European regulation that is being changed as we talk, this is very much in line with this new European regulation on health technology assessment, but also with the FDA diversity guidelines for over the past few years. With that, thank you very much for your attention, and yeah, I appreciate very much this opportunity.
Thank you very much, Frank, for this very insightful presentation. So we have received a couple of questions. Before we move on to the question, we actually have Cornelia who joined the webinar. Cornelia, you have been promoted to panelist. So if you want to say a few words. So maybe we can start with the question and answer. Yeah. She can jump in. So first question that has been asked.
Excuse me.
Go ahead,
can you hear me now? Okay,
yes, we can hear you. Yes.
Yeah. No, thank you very much. I personally, I found very important many points Frank mentioned, but for example, this concept of broadening the population, it's very important. I want to say that the core RCT structure that is kept. So if you start to do only pragmatic trials, and we have examples, what happens is the magnitude of effect that goes down, that is being reduced, and you need much larger studies. If you do them, in fact, pragmatic studies in the future only, I have heard these proposals. So here, the structure that you keep the RCT, it's very important.
Thank you, Cornelia. So we can ask you some questions, Frank. So first one, for the FACTIVE design, how would we adjust for multiplicity when using this design?
No, thanks, Aaron. That's a good question. Now, I would see it as that the analysis will have to be like, so the idea of this such an augmented phase 3 trial is that you're answering different questions from different agencies. So therefore, the data we are collecting, the evidence we are collecting, and therefore the analysis that has to be aligned with all this, they have to be directed towards the agency for whom you're answering the questions. So that is to say that I would not adjust for multiplicity across the questions for reimbursement and regulatory agency, but the protocol or the sub-protocol for the RCT part for the regulatory agency, that remains the same. For that, you have to then adjust for multiplicity, whatever the regulators want. Now, the questions for the reimbursement agencies, that's a different set of questions because that's a different discussion. So I would not mix up the analysis in that sense, if that makes sense what I'm saying.
Thank you, Frank. Second question from Aaron. Also, could we consider the FACTIVE design as a form of adaptive design where we are taking into account the real-world conditions?
Yes, indeed. The way how I presented it with the interim analysis, but it's not about using the real-world data in your actual randomized control trial, but that's just one way to trigger this part B. And the part B is a separate part of this under this one protocol, but that's a separate part, so to speak. So it's not about using this real-world data for the ongoing RCTs, but it's to generate additional data as needed in this part B. But yes, the way how I presented it with the interim analysis, yeah, that you can call this an adaptive design, yes.
Great, Frank. I will ask the next few questions. So a question from Natasha Muman, who wants to clarify the difference between a non-interventional pragmatic single arm in this RTC from standard of care arm in a conventional RCT.
Yeah, so I was probably a bit quick, but in this augmented design, we have different levels, right? We have the broader population. So there are eligibility criteria like RCT eligible criteria in patients who are not RCT eligible. And then you're having the treatment conditions. They can be given under RCT conditions or under real world conditions. So in a regular RCT, you would then randomize patients, RCT eligible patients, to either receive treatment, the new treatment, or control under RCT conditions. But that was not feasible. So the idea was therefore to break this thinking. Okay, we can randomize patients that are RCT eligible, but we receive the control arm under real world conditions. And under real world conditions here would mean that I was a bit quick here on the slide, but of course we only go to specialized centers, and that would be following the physician's discretion, so to speak, and they would probably, in all likelihood, then treat those incoming patients using these just approved CAR T therapy. So CAR T therapy is under the real world conditions being applied to. Yeah, so I hope it's clear. Otherwise, we can always touch base afterwards.
Right. There are a couple of questions here that are related. So one is, what is the risk to include a broader population in the RCT? Will it delay the duration of the RCT, delay the drug approval more than the traditional RCT that includes a more narrow population? And I guess a related question from another attendee is, what is the risk of this design? Will it dilute the effect size and reduce the likelihood of approval?
Yeah, no, these are great questions. And yes, so and these need to be thought through. Now we received similar questions, of course, from our internal teams, and I guess there are a couple of answers to that. So for example, and all the questions are very valid and they need to be thought through. But if you also have heard, for example, from clinical operations, oh, great. That means during the screening process, we can much reduce the screening failure because we are allowed to also include some of the blue patients from the broader population, which we otherwise would have sent away. Well, that helps recruitment pace because it seems like, okay, our company is being perceived as, okay, we have lower screening failures. So not only you are not sending patients away, but actually you're becoming more attractive. That's some of our clinical operations people said that. So that is with respect to the danger of like prolonging the core RCT. Now, the core RCT itself will not be touched. It's the same sample size. And of course, we understand there's somewhat of a competition if you add additional RCT eligible patients, but you use them for the close to real world part. So we understand that part, but then there was this comment from clinical operations. So it has to be really looked into details, so to speak, of the specific project. As opposed to the danger of like having small effect size, yes. So that is something very real, and of course, if you paid attention, we would have staggered these two parts so that the readout of part B would only take place while you're having the conversations with regulators. So that, and you have, of course, you have to have this upfront agreement with regulators, what is exactly they need for their decision making, versus if you include other data and generate other evidence that this is for primary use by other stakeholders. So that needs to be considered in more detail. And I think in the paper, we have a couple of comments and paragraphs on this specific aspect.
Thank you, Frank. We have another. Oh, Cornelia, you want to jump in?
Yes, I wanted to comment, and just what Frank said is all very much correct. I just wanted to mention that this broader population, it can, for example, contain patients who have a milder form of a disease, and we cannot have an impressive treatment effect. But if you start to evaluate them with all patients together, which we would do in a pragmatic trial, for example, then, of course, you are going to see this effect as well. So therefore, I can only underline, Frank, it's very important that the RCT structure is kept, and that is the primary evaluation. Yeah, as Frank mentioned, and the conversation with regulators takes place based on the RCT, this RCT part, the core. All the others they give additional information. Yeah.
Thank you, Cornelia. So maybe that's related to the next and last question due to sake of time. Can you comment on some regulatory acceptability of the idea to run randomized clinical trial and pragmatic clinical trial in parallel? Do you think FDA, EMA could push back to protect study participants if pragmatic control trial is an additional risk?
Sorry, I didn't see this question, and it was a long question.
Yes. From Pierre Mancini, maybe.
Oh, here, Mancini. Yeah, okay. Yeah, so yeah, I think it was very brief to say, I'm not sure whether I fully understand this question, Pierre, but yeah, I was indicating that we would not include patients who, for whatever reasons, that they would be put into a safety risk. And of course, if you have a compound where you are first in market, then of course you have limited safety database, and then including those non-RCT eligible patients, you don't know whether they would still be eligible from a safety perspective. But so again, this is something that we also have discussed briefly in the paper. And maybe in one setting, it's more, the use of this is more amenable than in another setting. So that's I'm not sure whether I fully understand the question, but yeah, again, happy to take that offline. Mm-hmm.
Yeah, do you want to jump in?
No, thank you. I think my point is, sometimes in some regulatory interaction, I feel there is a general resistance to those ideas to move quickly, and in particular, I mean, that they want to have like safety checks or even effectiveness demonstration before we go into, less rigorous patient follow-up and monitoring. So that's a little bit the sense of my question. In theory, I love the idea, especially, working both in development and medical affairs. But in practice, I can sense, some of the FDA division at least would be kind of very conservative on the matter. So I wanted to kind of hear your thoughts about it.
Yeah. Okay. Now I understand it better. Thank you. No, and now I appreciate this. And I'm not saying it's easy to especially the entire scheme that I had shown. I mean, I would not envision to go this way, but yeah, but specific aspects can still help in specific settings, specific aspects of the overall framework. And again, maybe part of the answer is the same as the previous question. We need to have a clear agreement and understanding with regulators what is the information they need for their decision making, right? And this is the core RCT, and we are not going to touch on this, and we are not at a sudden reducing the sample size for the core RCT in favor of generating other information. That's not the proposal. The proposal is to augment the phase 3 part to generate additional information that could possibly be helpful for regulators, but is primarily also of interest to other stakeholders. And again, it really depends on the specific context, yes. And that is also why in this paper we had invited, I mean, it's really a joint collaboration with Rob Hemmings, who used to be a regulator, Anja Schiel, who is one of our leading reimbursement specialists here in Europe, or HTA body specialist in it. So that was very specifically why we had different voices in that one paper as a collaboration.
Thank you so much.
Thank you, Pierre. Thank you, Frank, for the answers. So that brings us to the end of today's webinar. So on behalf of the Boston chapter of the ASA and Cytel, I would like to extend a huge thank you to Frank for sharing his insight and expertise, with us today. I think it has been a truly valuable session for all of us, and we really appreciate your time and the effort you put into this presentation. So thank you. And I also would like to thank you all for attending and engaging with us. Your participation and thoughtful questions made this, enriching discussion. So as I said at the beginning of the webinar, a recording of today's session and the slides will be shared with all registered participants. So please keep an eye on your email for more details. And we also would like to encourage you to stay engaged with the Boston chapter of the ASA. So don't forget to check out our upcoming events, visit our website, or follow us on LinkedIn to stay updated on our future programs and networking opportunities. So thank you again for joining us, and we really look forward to seeing you at our next event. Have a great rest of your day. Thank you.
Thank you all.